Hi guys, welcome back to the channel. In this video, I'll be carrying on from the last video and we'll be looking into binary fission and HIV replication, and this is also from the all cells arise from other cells topic from the A-level biology curriculum. So let's begin. Okay, so now looking into prokaryotic replication by binary fission. So as we know that the prokaryotic cells have circular DNA as well as plasmids, which store the genetic information. So in this, the circular DNA and the plasmids would replicate first, and this happens by the semi-conservative replication. So remembering semi-conservative replication, so we would have our one original DNA as well as forming one new DNA, and this would be our semi-conservative replication. So as we can see here, we start with this prokaryotic cell, and then when the semi-conservative replication occurs, we end up forming two circular DNA strands, and we can have a variable number of plasmids when the replication has occurred. So what happens next is the cell would enlarge in size, so it'd get bigger, and each DNA molecule we have here would start to move to the opposite pole of this cell. So if we look again, so as you can see, the cell has gotten bigger and the DNA molecules have started to move to the opposite poles of the cell, as well as these plasmids. So now this is similar to the anaphase stage, which occurs in mitosis, which we've looked at. And then finally, to finish it off, the cytoplasm would divide into two daughter cells with a single copy of circular DNA. So each cell that would form would get a single copy of the circular DNA, as well as the variable number of plasmids. Okay, so looking at the diagram now again, so as we can see that we have formed two new cells, so the cell walls aren't connected anymore, and these would be our two daughter cells, each containing a single copy of the circular DNA, variable number of plasmids. There can be more than one plasmid, it does not have to be one, and that's why we tend to say variable number of plasmids. And this would be the same for the other cells. So we've formed two daughter cells. Now looking into the replication of viruses. So you need to know that viruses don't undergo cell division. And now this is because they are non-living, so they can't carry out, for example, mitosis or binary fission. So they require a host cell, and they would hijack that host cell, and we'll be going through the process now. So what happens first is the viral proteins would bind to the receptor on the host cell, and the viral membrane and the host cell membrane would fuse together, releasing capsid. So looking at this diagram, so we have this virus, and these little purple dots on it are representative of the viral proteins. So the viral proteins would bind to this cell. So in this cell there are many receptors, and the viral protein would bind. So this purple protein is going to bind to a receptor on this host cell, and then the membrane, so the viral lipid membrane, as well as the cell membrane, the phospholipid bilayer, would fuse, and this would cause the virus to enter the host cell. So as you can see, this virus has entered, and the virus would release its capsid. So next what happens is we have an enzyme. This enzyme is called reverse transcriptase. So what it does, because the virus has a viral RNA, it does not have DNA. So what this enzyme does is converts this viral RNA to complementary DNA, because in the nucleus of cells we would have DNA, we don't really have RNA. So what this does is converts this viral RNA and to complementary DNA, and now this DNA can be joined to the host cell's DNA. So there's an enzyme involved which is called integrase. Now this would combine the viral DNA to the host cell DNA. So it basically joins the two DNAs together. And this new DNA, so this new DNA which contains the viral DNA, would now make viral RNA. So as you know, through transcription, what happens is the DNA is converted into mRNA, and this is what's going to happen now. So the DNA is used to make viral RNA, and this viral RNA is of the virus that's infected the cell. So as you can see in the diagram, we had this virus which released the enzyme reverse transcriptase. It released its viral RNA, and this viral RNA was converted into DNA, and now the cell replicates and it produces the viral RNA now. Now next, the viral proteins would be made at the host ribosome. So using that RNA and that's been produced by the host cell, the viral proteins which it needs, for example, the proteins on its surface to bind to the cell, and other proteins would be made at the host ribosomes. So as you can see in the diagram now, so we have produced these proteins, and now what would happen is the assembly of the new virus particles would occur. So all these proteins, all the stuff that's made of the virus would combine together now. So the assembly of the new virus particles is occurring. So now we have just formed this virus, and it's just everything that it needs, just without its outer membrane. So finally what would happen now is the virus would bud off from the membrane. So what budding off means, it would just basically steal the host cell's membrane. So if we're looking at the diagram, so it would steal this membrane, so all this membrane, and it forms the virus which was originally there. So as you can see, both of these viruses are the same, and by one host cell, when this virus infects it, we can be forming so many viruses, and what would finally happen is the cell would be destroyed. And this is basically the replication of viruses. Thank you so much for watching this video. If you did like this video, don't forget to subscribe to see more of these, and you can watch my recent videos by clicking on the links popping up. Thank you.